Good day all. I was recently unable to attend a virtual group meeting after suggesting we read and discuss a paper and have journal club, and it was semi-seriously suggested by the boss man that I give a vlog presentation of my group meeting. So now you get to learn all about this fantastic study in NSMB from about 2 years ago on yeast surface display and nanobodies. Wonderful! Okay, so a bit of background to antibodies and nanobodies. So here we have a schematic of an antibody, which is the thing that affects the adaptive host immune response, and it's composed of a heavy chain and a light chain. Also two heavy chains and two light chains, which are disulfide bonded together in this Y-shaped structure, and the antigen binds at the very tip between the light chains and the heavy chains. The reason for this is that this bit is variable, so this is what gives specificity for the foreign agent, and this bit stays constant and binds to the surface of some immune cell to mount a host response. So a good example of this here is immunoglobulin G, this one on the left here. So we have this heavy chain here, constant region, variable region, constant region of the light chain in blue, variable region of the light chain. Thing binds up here, immune response mounted. Great. So this is from Ablink's company that obviously produces nanobodies. So the difference here is that on the left here we have an antibody. It's a very large structure, but on the right we have a nanobody, which is from a camelid, so a llama. And these have evolved a different structure, such that they have the constant regions, but they don't have this large arrangement of 4 different domains at the top here, whereas they just have one variable region which gives specificity for invading antigens. Sharks also have this for some reason. I didn't look into it, but obviously to get antibodies you need to put a foreign agent into an organism which will then mount an immune response, and it's probably a lot easier to deal with immunizing llamas than it is with sharks, as llamas live on land and also they don't want to kill and eat you. So why would we want to make use of these nanobodies? One example is in the structural biology of G-protein coupled receptors, a family of transmembrane receptors which are very important in responding to extracellular stimuli and activating downstream signaling pathways. And these are a massive drug target. There are 108 of them currently targeted by drugs, and the sales value of this as of 2018 was $180 billion. So here's an example from Brian Kabilka, the Nobel Prize winner's lab, of one of the adrenaline receptors. And so one of the reasons you would want to do this is because an activated state, especially in this case, requires the binding of a small molecule which can dissociate, and so you get left with a heterogeneous selection of things in solution, which for protein crystallization, when this work was done, was not good. You need something homogeneous. So here, in this figure panel 1A, you can see a structure of the GPCR transmembrane region, with the nanobody stabilizing it down on this side and a small molecule bound up here. So it can help us capture these states, and you can see quite clearly it's different from the structure in blue, which would be the inactive state, I expect. And so often when people have worked on the inactive states, that non-ligand bound stuff, they've required some sort of mutation to capture it in that state, which of course is not physiological, or is physiological only in the disease state. So this way, with the nanobody, we can see something much closer to the real physiological case of the protein. The other reason is that antibodies are very large, lots of disulfide bonds, and they require, and they're composed of separate chains, so they require extensive production, generally using HEC293 T cells, which is expensive, time consuming to get a lot of protein. Whereas nanobodies, the chain is just a single chain at the top that constitutes the affinity for the antigen, whereas this is at the top of both variable heavy and light chains, domains even. So we don't need to use HEC cells or complicated protein expression systems to make these nanobody variable regions. We can do it in bacteria. We can just send it to the periplasm. This is much cheaper and generally produces a lot more protein. So back to the problem at hand. Obviously, since these were discovered, it's been noted that they have enormous potential for at least structural biology, and maybe as drugs, but we haven't quite got there yet. But a bottleneck, as stated here, have been obtained. It requires animal immunization, which takes a lot of time, is expensive, and can produce quite a massive number of nanobodies. I knew somebody who did this and got back something like 60 or 70 different sequences for nanobodies when she subsequently had to purify all of them and test all of them for binding to see which was the best one, because that's when you would scale up for at least in her case doing structural biology. So here this paper points out a way of basically they create a massive library of nanobodies, and they use what's called yeast display to screen all of these simultaneously and progressively remove the weak ones from the system until they've just completely enriched with the tight binders for the target in question. And so this has hopefully got broad applications both in structural biology and drug discovery, and maybe in some other sort of biotechnological tools. So the final question we have to discuss before dissecting this paper properly is, what is yeast display and why do we use it? Why is it better than using the llamas themselves? Because whilst time consuming, the llamas will give you a nanobody. But one of the problems with the llamas is that if there is a conserved epitope of some sort between the protein that you want to raise a nanobody to and one of their proteins, this will be removed from antibody selection by the llama's immune system for fear of raising some sort of anti-self immune response, which obviously the llama does not want. So instead we use yeast display. In this case we have a genetic library of constructs which contains a whole number of variants of our protein of interest, this thing in green here. The example in our case is a nanobody. This is genetically fused to a protein called AGA2P, which is a yeast protein, which will bind it to a cell surface exposed protein called AGA1P from the yeast, which is covalently attached to the yeast cell wall. There are other tags genetically encoded in this case, an HA tag and a MIC tag, and these are just used for detection that the thing is at the cell surface, and this has been shown with antibodies with a green label. So the way that we would go about this process is to use something called fluorescence activated cell sorting. So that's a method via which you can use fluorescent signals to separate cellular populations from other cellular populations. So the workflow in this case would be that we'd have our pool of nanobodies exposed at the cell surface of the yeast. We would then add our target protein. So that's the thing that's shown as an antibody here, quite confusingly. The thing that we want to generate a nanobody for. That has some sort of fluorescent tag on it, and we use the fluorescent activated cell sorting to separate the things that don't bind the fluorescent signal, i.e. the tagged protein, the tagged target protein, away from things that do. And very rapidly we can build up a pool of nanobody constructs that have high affinity for our protein of interest. Why would we want to use yeast display instead of phage display? Phage display being the most commonly known one. Two reasons, mainly because yeast have more convoluted folding pathways, so give us the ability to do more targets. Nanobodies have disulfide bonds, so we wouldn't be able to use phage for that. And also fluorescence activated cell sorting, keys in the cell part, so phage would not be amenable to this. So, figure 1. Here we have a general schematic for a nanobody. Primary sequence, N-terminus to C-terminus. And there's a structure shown in figure 1b. So the structure of a nanobody is similar to that of a fab fragment with a bunch of beta sheets, and they have 3 complementarity defining regions, the bits that bind to the target protein. So in this paper what they did is they took all of the structures of nanobodies they could find in the PDB, so there were 93 when this was published, and studied excuse me, all of the complementarity defining regions. And here in figure 1a they have shown the residues that tend to be conserved in these positions in each one of these loops. Shown, for instance, the second position in complementarity defining region 1 is an asparaginous, serine, threonine, or a tyrosine. But in these positions here, the asterisks, they're variable. You see the similar things in regions 2 and 3, and the other thing about region 3 is that it can be of more variable lengths. So they went through and based on popularity of certain residues in certain positions, they generated a library with variable residues in the positions of the asterisks in each one of these. And using a very, very convoluted PCR method, they were able to generate a library that we predicted would have this diversity. Figure 1c shows a graph after sequencing, so they had some 480,000 sequences analysed. They predicted that they would have roughly 100 million different sequences, and within those sequences, I assume selected at random, they saw that the frequency of amino acids within the positions in the complementarity defining regions correlated quite well with what they had designed it to be. The group subsequently selected at random 11 nanobodies just to scale up and purify from periplasmic expression in E. coli. 9 of the 11 behave quite well after running through a size exclusion column and look relatively monodisperse. I think these gels are just from after the his trap column, and they're already pretty clean just from periplasmic extraction. Back to figure 1. Figure 1D is a schematic for how they engineered their nanobody library to be stuck to the yeast cell wall and presented. And instead of using AGA2P, as I had talked about earlier when discussing how yeast display works, they just used a very long linker that would get stuck to the cell surface and an HA tag to make sure it was there. How did they do this? They designed a whole load of variable lengths, and they used fluorescence activated cell sorting. So the key part to how this works is that they took a known complex between these proteins CV1 and CD47 and stuck the CV1 at the cell surface using linkers of different lengths, and then used fluorescence activated cell sorting against the HA tag, and by adding the target protein CD47 with a different coloured label on it. And so this is an example of fluorescence activated cell sorting. So in this dimension we have cells sorted on the basis of anti-HA, so the region between the linker and the protein of interest fused to the cell surface. And on this axis we have CD47 biotin. So what they've done here is they've used a CD47 protein with a biotin tag on it and then added fluorescently labelled streptavidin. And the key to this is they use a different colour, so it's a wavelength 647 nanometers, to the Alexa at 488 nanometers, and so they separate in 2 dimensions, and the populations of cells are shown as black dots. So obviously this correlates with how well exposed the CV1, i.e. the target protein fused to the cell surface, is. And they show that this top quadrant here, where they get good labelling of both the HA tag and the fused protein, is with a 649 amino acid linker. So that's what they go on to use for all of their nanobody constructs. And so finally in figure 1E they have a schematic for their experimental workflow. So we start off with a library of yeast, each cell expressing a different nanobody fused to the cell surface with this long linker. They add in their antigen, so the target protein for the nanobody with some sort of tag on it. And so they will then use something to separate away the tagged yeast from the untagged yeast. So the non-binders separated via, for instance, fluorescence activated cell sorting, and they get different populations where they have the fluorescent cells. They can also use magnet activated cell sorting, which could be a little bit faster but less selective, where they use a magnetic bead that binds to the thing on the protein of interest, and then they just wash off all the other non-binders, and they get an enriched library of binding nanobodies, and then they amplify those specifically, so only the ones that bind to the protein of interest are expressed, and then just repeat this and enrich until we have very, very few nanobody sequences left, sequence, find the thing that binds, and then purify the nanobody in question and characterize it. So now that they had their library set up, the authors of the paper decided to screen for a nanobody that could bind to human serum albumin, HSA, the most abundant protein in human plasma, an important drug target. And using a combination of MAX and FAX, they enriched a pool of nanobodies, and after 4 rounds they saw that HSA bound more tightly to the population of cells that they had, as shown by this blue next to this black trace. And sequencing showed this particular nanobody, B201, was enriched within that pool. They scaled up and purified said nanobody, and here's a quick size exclusion trace in figure 2b to show in red they've mixed HSA with nanobody B201. In the dotted trace is just HSA, and in the dashed trace is just the nanobody. And so they show quite nicely from gel analysis of these peaks that peak 1 of this trace in red contains both HSA and nanobody 201. So they have formed a complex and verified that it binds. So they go on to analyse this binding via SPR, and they titrate over an increase in concentrations of nanobody B201. Shows an increasing response as we would expect from an SPR experiment. And importantly, with mouse serum albumin, so MSA, they see absolutely no binding, so no response in flowing over their nanobody. So the interaction is specific for the human variant of this albumin. So in figure 2c, the group go on to crystallize the nanobody in complex with HSA, and they are able to solve structures of the unbound nanobody, it's not shown here, it's in the supplement, and also the nanobody HSA complex. And they see that complementarity defining regions form contact, all three of them form contact with the HSA, and the recurring feature is tyrosine glutamate hydrogen bonds. And then they see quite large changes in the conformations of these loops, so it's the grey to the coloured, so this moves in quite far, I don't say how far, in order to form a complex with HSA, and this is verified quite nicely with electron density. So in supplementary figure 3c, we have the density for the bound nanobody, and we see this compared to, in grey, the model for the unbound nanobody, and we have the density for the unbound nanobody in supplementary figure 3d. So we see that this loop especially moves in quite drastically, as well as this tyrosine 33 flipping out to a different conformation as a result of binding. And the authors also go on to talk about how they're able to go from having just this library and HSA to having this bound structure to a nanobody within, I think, 2 to 3 weeks. And as you can do this using MAX, which just requires the use of magnetic beads and a good experimental design, one can do this without even needing a fax system, which is more expensive than a pot of magnetic beads. So an interesting piece of work that the authors of this paper did that wasn't in the main manuscript, was in the supplement, is raising nanobodies using their yeast library against a heterogeneous mixture of proteins. So in this case they use an example protein called adiponectin, which is reportedly awful to purify and aggregates so badly that you can't break apart the aggregates with SDS or boiling, but is apparently, they don't show a blot, but apparently is quite happy in this complex milieu of all the other stuff around it. And so here's an experimental design here. Well, basically they've got a load of contaminants, but they use the yeast cells to probe for adiponectin binding. The adiponectin has an antibody tag on it, and they use an anti-flag antibody with a fluorophore on it to find out the cells that have got adiponectin at the surface. And so using FACS, they demonstrate this quite nicely, that with the adiponectin and anti-flag antibody, they have a shift towards higher fluorescences in this cellular population over here. They sequence a bunch of nanobodies out of that and see quite a lot of variety in the complementarity defining regions. They go on to do a pull down of the adiponectin in the absence and the presence of one of the nanobodies. So here on the left they have a blot against the adiponectin, presumably against the flag tag on it, in the heterogeneous mixture. See that it's nice, but as soon as you try and pull it down in this column here, it's basically not there. So the pull down process is very difficult, probably owing to the poor solubility of this construct. When they have the nanobody present, they pull down a great deal more adiponectin. This nicely demonstrates that this nanobody binds. And then they take one of the nanobodies and do SPR on it, and they see increasing binding response for a titration, indicating that indeed it does bind. So at this point we go into the real meat and potatoes of this paper, a nice proof of principle. So what the authors sought to do was to show that their library could be conformationally selective, and so once again they use the great Kabilka example of the beta-2 adrenergic receptor, which we know has been crystallized with an activating nanobody. So in figure 3a here we have a little schematic that shows that there is an activated state, which they've coloured green, and an inactivated state, which they've coloured red. So this corresponds to two different ligands bound to this purified GPCR in the form of an agonist or an antagonist. So what they do before they do all their sorting experiments is take purified protein that's got a flag tag on it that has been mixed with the agonist or the antagonist. So figure 3b shows the 3 of the rounds of their sorting approach via FACS. So in each case they've mixed the yeast population with either the beta-2 adrenergic receptor and agonist, which has been labelled with Alexaflur 647, or beta-2 adrenergic with the antagonist, which has been labelled with Alexa 488. And so they sort on the basis of either one of these two colours. What they want to see is staining in the far right quadrant here, because that would imply that they were just getting signal from the Alexa 647 that was bound at the yeast surface. And so they're using this to try and pull out something that's just responsive to the agonist conformation of beta 2, which they have successfully done by the end of round 6. 3C shows their approach is quite advanced. They use max, 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 and then fax twice, and max, and they lower the concentration of the added activated protein to try and increase the affinity of the thing they're pulling out. But they do counter selections before in each case to reduce the populations of nanobodies which bind to the inactivated conformation, i.e. the antagonized one, or one of the antibodies used to stain the flag-tagged activated version. So after enrichment, they pull out this whole pool of nanobodies, which has presented them with the issue I told you at the start: yeast display would circumvent, but at least it's not 60 nanobodies, where they've got a whole range of sequences in complementarity defining region 3. And so their approach to first whittle down which one of these are worth scaling up to do binding experiments with is to use an on-yeast titration experiment. So in supplementary figure 5, what they've done is they've expressed just the nanobody in question at the cell surface from whichever clonal population they have, and they've added progressively more and more and more of the activated beta-2 adrenergic receptor with the agonist, labelled with a particular colour, and then measured the amount of cell surface fluorescence. And so they've used that, after they get these sigmoidal binding curves, to imply a sort of KD, the EC50, to get the things that bind the tightest. And so the lowest numbers are seen for these ones, 200 through 206. So those are the ones they purify. They choose C200 to C203, and they use a radioligand competition assay with a purified beta-2 adrenergic receptor and the purified nanobodies. So the black trace here is for no nanobody. So what they've done in this experiment is they first bound the beta-2 adrenergic receptor to a radio-labeled ligand, so this is that tritiated DHA, and then they've added progressively more and more and more adrenaline, which is a ligand for obviously the adrenergic receptor, and they have observed how much radioligand is still bound. And in the case of all of these, they shift the curve to the left, implying that it takes less adrenaline to compete with the DHA binding and stabilizing the activated conformation. They go on to characterize the interaction in vitro using purified proteins on an SPR chip, so single cycle kinetics. And so they run these 4 nanobodies they purified over and get varying affinity values. This is comparable to the value of the nanobody used in the Kabilka paper, so these are all roughly the same or tighter. They then use nanobody C200 for some reason not mentioned in a pull down, figure 3H, so that has a his tag on it, and they've used beads to pull down the nanobody, and it brings out the beta-2 adrenergic receptor, which has a flag tag on it and no his tag, so they form a complex in solution, and they get some nice protein crystals in lipidic cubic phase crystallization of the two. I hasten to add that these are the first crystals in the presence of this agonist BI167107. So in figure 3J, they seek to define the application of these nanobodies in a cellular environment using a cyclic AMP signaling assay. So in this experiment, they use cells transfected with the beta-2 adrenergic receptor and the nanobodies in question, and in each case they show that the EC50 value in response to increasing adrenaline concentration is decreased by the nanobody. And they also highlight that it's important to screen a number of these nanobodies in this sort of setting with biotechnological aspirations, because despite having a lower affinity than at least 2 of the other nanobodies, it shows a larger decrease in the EC50 value. This is likely because in this experiment they're resident in the reducing environment of the cytoplasm, which would be destabilizing to their fold in general as they have disulfide bonds. So there is also an element of nanobody stability to consider when conducting experiments for things other than just pure structural biology. So in this final figure, figure 4, the authors apply their approach to a new problem, the adenosine receptor, A2A, because whilst it is great to use the beta-2 adrenergic receptor as a proof of principle, this has already been done. But, this adenosine receptor, which is important in the nervous system and also is apparently a hot target for cancer immunotherapy, has no active state stabilizing nanobody. So they use the same approach here in figure 4A of using an agonized conformation of the receptor with this colour, and again an antagonised version with a different colour, and sorting the stained yeast correspondingly. And again, what they want is to find stuff that sits in this quadrant here, so stained by just the agonised conformation of the adenosine receptor, again implying that there are nanobodies binding to that at the surface of the yeast. And after their sorting approach, after successive rounds, that's what they see. And taking specific nanobodies from these yeast populations, they restain and see that the entire cell population is stained just by the agonised adenosine receptor. They then go on, like in supplementary figure 5, to use a staining approach for yeast cells, where the nanobody is presented at the surface, and they stain with either the agonised or the antagonised conformation of the adenosine receptor, and it selects quite nicely for the agonised conformation. And in figure 4D, they've scaled up these two nanobodies which select for the agonised conformation of the adenosine receptor, and done pull downs as they did before with the beta-2-adrenergic receptor and nanobody C200, I think it was. And in this case, the nanobodies only pull down the adenosine receptor in the presence of an agonist and not in the presence of an antagonist, implying that indeed the nanobody sorting process has selected for nanobodies that just bind to the agonised version of the adenosine receptor. So to sum up, yes, the authors of this paper have designed an enormous nanobody library with clear selectivity for multiple different proteins, be soluble proteins or membrane proteins, and they have shown quite elegantly how one can use this to acquire a nanobody which stabilises the desired conformation of your protein. This could be excellent, not just for structural biology, but for finding sensors for things, for pulling stuff down out of environments, and then ultimately combined with structural biology, drug discovery. And so whatever targets that do still exist that have eluded us as structural biologists, we can tackle absolutely, especially with the heterogeneous mixture approach as well. This will really help us to find ways of producing those and solving their structures. And so here we have a publication that's a direct follow-on from said nanobody work. So in this paper, the authors selected a nanobody for the activated conformation of smoothened based on its agonist bound state with this molecule SAG, smoothened agonist molecule, and they found cholesterol molecules bound in the extracellular and the transmembrane regions of smoothened. This is very biomedically relevant because smoothened is the activating protein in hedgehog signalling and is indicted in a whole load of cancers as well as organism development. And up to this point, the structure of a wild type active smoothened had eluded us, and it was gained using this nanobody based approach. And so here it is applied in structural biology and also maybe in the future drug discovery, and that would be excellent.